Cell Structure Part 2. In the late 1600s, around 1660 or so, there was a scientist in England by the name of Robert Hooke. Robert Hooke was a very prolific scientist, and in fact today we still use some of his mathematical calculations in engineering and physics. But one of the hobbies, if you will, or one of the extra activities that he endeavored in the 1600s was the upcoming new technology that's called microscopes. He improved upon what had been used at the time in the form of a microscope, which is illustrated with the Hooke microscope image on this slide. Now, it doesn't quite look like the microscopes that we use today, but it does have the fundamental elements or parts that we do expect to find in what we would call a bright field or light microscope. There's an eyepiece or ocular. There's a barrel that holds magnifying lenses. There's a means of focusing the specimen or the image, and what we call the objective magnifying lens. There's a place for a specimen, which of course is very different from the microscopes that we use today, because the specimens that Robert Hooke viewed through his microscope were macroscopic to begin with. He simply used the microscope, which was one of the sort of hobbies, if you will, that folks were using at the time with magnifying lenses, is they would take magnifying lenses like magnifying glasses, or in this case these microscopes, and look at items that you could see with your unaided eye, but you just couldn't see a lot of the tiny detail. You illuminate that specimen somehow, and this illustration on the image in the photo has the use of an oil lamp, and you'd see the tinier details of that structure. One of the items that Robert Hooke viewed through his microscope was a thin slice of cork. Cork from the bark of a cork tree. Now, real cork is a very spongy material. The reason it's spongy is illustrated in the image on the lower left, which is an illustration or drawing from Robert Hooke's book about the things he saw using his microscope. All of those little black holes or little black squares or rectangles in this image of these thinly sliced pieces of cork are empty spaces. So that the remaining structure, which is illustrated sort of in a beigey white, is essentially trapping all these little holes. That's what makes cork spongy, is it has a lot of empty spaces in it. When Hooke saw these little empty spaces through his microscope, he said they looked like honeycombs or bubbles, or one of the other descriptions that he used to describe what he saw when he looked at this thin piece of cork was cells, meaning small little rooms like a monk's cell, or nowadays we would think of it as a prison cell. We still use that term cell today in a variety of ways, including the fundamental unit of an organism called the cell. And this is where we get that term, from Robert Hooke's description of these spongy, bubbly structures that he saw in this thin piece of cork. For whatever reason, that concept or that term caught hold, and we still use it today after several centuries. He published his observations and his discoveries in 1665 in a book, and we still have copies of that book and of course copies of the illustrations of what he saw through his microscope. We jump forward about 150 years, and we have lots of scientists who of course are using microscopes and using other structures to study these tiny little things that we can find in plants, animals, fungi, and even drops of pond water. It took Theodor Schwann, who was a zoologist studying animals, and Matthias Schleiden, who was a botanist studying plants in Germany, two separate towns or two separate cities in Germany, and at roughly the same point in time in 1839 to propose the concept that all things were made of cells. Because when Schwann and Schleiden took a look at body organs and tissues from their various specimens of animals and plants, they could see that the fundamental units of all of those animals and plants were cells. Now, at the time, Schwann and Schleiden really didn't know where the cells came from, though we take it for granted today that we know that one cell produces another cell. That actually took a different scientist by the name of Robert Remak and then Rudolf Virchow to further that concept of where cells originated. But we still always give Schleiden and Schwann the credit for what we now call the cell theory, that all living things are made of cells. And that is one of the general principles or concepts in biology. A theory in biology is a concept that first starts out as a question that can be tested or evaluated. We call that a hypothesis. A scientific hypothesis is a very particular kind of statement. It's a statement that's basically a question that we want to find an answer. And so it's testable. We have to be able to set up an experiment or a procedure or some way to determine what's the answer to that question. And so after evaluating many specimens, it was universally agreed to that cells are the basic units of organisms. And what they're thinking here are plants, animals, fungi, algae. And eventually we even get down to single-celled organisms, so that all organisms are made of cells. All cells come from pre-existing cells. That too was a concept that was eventually validated. And so once we've validated or answered our question or our scientific hypothesis many times, and we're certain of the answer, that yes, we get confirmation of whatever that discovery is, then we generate what's called a theory. And so a theory is a well-supported, well-tested concept. Now, there are two types of cells found on Earth. We call them prokaryotic and eukaryotic cells. The one fundamental difference between prokaryotic and eukaryotic cells is the presence or absence of a nucleus, a well-defined structure. Prokaryotic cells do not have a nucleus. Pro means before. Karyo is referring to seed or kernel, meaning this little thing that we'd expect to see inside of some types of cells. And prokaryotic cells do not have a nucleus. Eukaryotic cells, on the other hand, eu meaning true, karyo again referring to seed or kernel. So these cells do have true structures that we'd identify as a nucleus. Now, as it turns out, in the 20th century, entities were discovered that are called viruses. The existence of viruses does often challenge cell theory, because microbiologists have no trouble calling viruses microorganisms, but a classically trained biologist who uses cell theory as a overarching underlying principle of biology would find it difficult sometimes to describe a virus as an organism, because viruses lack cell structure. So concepts do arise that challenge theories in biology or in science. And whenever those challenges arise, then it's up to scientists to address or answer those challenges. So these are constantly evolving or changing concepts. They're not written in stone. You have to constantly question and constantly evaluate. any scientific theory. Cells do have common components, and that's what this chapter is all about, the common components. Cells are going to have some kind of outer barrier. We call it a cytoplasmic membrane. It can also be called a cell membrane or plasma membrane. It'll have internal gel-like material. It's mostly water, but also suspended or dissolved biochemicals. It's called the cytoplasm. We can also use the term cytosol to refer to the cytoplasm, gel-like material, and the water and all the components. We have DNA and RNA. Those are going to be found in all types of cells. We have ribosomes and proteins. There are other types of structures that we expect to find as well, and that's what we'll talk about when we get through all of the parts of Chapter 4. Again, the two fundamental types of cells found on Earth, pro and eukaryotic cell structure. Then, in addition to no nucleus, prokaryotic cells have no membrane-bound organelles. We do expect to find membrane-bound organelles in eukaryotic cells. Most prokaryotic cells have a cell wall. The cell wall can either be made of peptidoglycan or pseudopeptidoglycan. Scientific evidence supports the concept that prokaryotic cells are the original cells on Earth. They appear to have developed on Earth roughly 3.6 billion years ago. The Earth itself is a little older than 4 billion years, so it's well understood or well accepted that the formation of cells developed shortly after, relatively speaking, in geological times. It wasn't too terribly long after the Earth itself cooled and formed that cells started to take shape. We have two scientific classification groups where we would find prokaryotic celled organisms. We call them domains. And one domain is called archaea, the other domain is called bacteria. Most students have heard about bacteria. You hear about it on all kinds of commercials on television or in the news or in magazines. Archaea, maybe not so much. Those are the types of prokaryotic celled organisms we find at the bottom of the ocean, around and near deep sea hydrothermal vents, or in very extreme environments on the surface of the Earth. Regardless, archaea and bacteria are two categories, or major groups, of prokaryotic celled organisms. For the generalized structure of a prokaryotic cell, we are going to use this illustration. If we take a look starting at the outside surface, a motility structure called a flagellum can be found on some types of prokaryotic cells. Not all, but some. You could have a single flagellum, as in this illustration, or you could have multiple, which is flagella for plural. We also find these hair-like structures sticking out of the surface. Those are called pili. They can also be called fimbriae if they're for attachment. And some prokaryotic cells will have these little attachment structures and some won't. That is something we might find. As we're going from the outside in, we might find a structure called a capsule. A capsule may be found on certain types of prokaryotic cells to give the cell extra protection and extra structure. In addition to a capsule, we might find a cell wall. In fact, many types of prokaryotic cells will have some kind of cell wall material that's either peptidoglycan or pseudopeptidoglycan. Just beneath the cell wall, if it is present, we're going to have the cell membrane. That's the fundamental outermost barrier of every kind of cell that we'd expect to find. And then if we take a look at the interior of what's on the inside of the cell, we're going to find these tiny little dots that are called ribosomes. And then a supercoiled or extremely tangled structure that we call a chromosome, which is where we find the material for hereditary information in the form of DNA as a chromosome or nucleoid. Nucleoid is the term that's used to describe the location of all the genetic material in the form of a chromosome in a prokaryotic cell. For some prokaryotic cells, it's not the only source of DNA, but is the principal or major source of hereditary material for the organism. For prokaryotic cells, size does matter. The small size is an advantage because smaller cells have a higher surface area to volume ratio, which is illustrated in this cartoon drawing on the bottom of the slide. Notice that what happens when you increase the size of the cell, the surface area to volume ratio decreases, which means it's more difficult for materials and food to get transported into the cell and reach the center of the cell. The smaller the cell, the higher the surface area to volume ratio, the easier, or we have an increase in passing material into that cell to get to the center of the cell. The larger the cell, the smaller the surface area to volume ratio, which actually decreases how easily materials and food and water could get transported to the center of that cell. If we take a look at prokaryotic cell structure internally, what we're going to find is because of this small size and the ease in which materials can get transported into and out of the cell to the center with not too much distance in between, we don't need a lot of what we call transport mechanisms inside that cell. Very nicely passive mechanisms will allow nutrients and water to pass in and out and get into all of the cell that's needed. With larger cells, like plant and animal cells, with that very small surface area to volume ratio, then the cell does need some assistance internally to transport materials all around the cell. And that's going to allow us to see why we have different internal structures. A lot of it has to do with cell size. The larger the cell, the more complex the internal structures will be, in large part because of this change in surface area to volume ratio.